So welcome, everybody. We are almost ready to begin. Okay, I think we're ready to begin. So good afternoon or good morning to everyone. I am Paula Garcia from ECRIN, and on behalf of the EULAC PerMed project, I would like to welcome you all to this webinar, which will focus on in-silico models and personalized medicine. We would like to thank the two speakers for joining us today. We are very excited to listen to your talks. And I will now hand the floor over to Dr. Manuel Barral Neto from Fiocruz, who will introduce both of the speakers.
So good morning, good afternoon to everybody. And it's really a pleasure to introduce the two speakers that are really, outstanding persons in their fields. And Anamaria Camargo is a doctor she works heavily on oncology, and it's really a pleasure to have her with us. She's has contributed a lot to the field, several publications, important publications in the area, and several awards that she received for her work. And it's really, she's one of our leading experts in the area, and in a very strong institution in São Paulo. So it will be a pleasure to hear Anna's contribution. And the colleague from German, Dr. Kirschner, Marc Krischner, is also a very outstanding person in these areas. He also had, got his PhD in Germany, and had for some time in the US for postdocs and work. And he worked in virology and immunology at Würzburg upon returning to Germany. And he's the scientific officer of a very important project for us that is really the project on data integration that is critical for personalized medicine, and it's critical for international collaboration in personalized medicine. So it's two complementary kind of views that we're going to have today. One focuses mainly on the aspects in oncology, and then a more broad presentation on how to use and integrate data. And this is going to be a very interesting seminar. So thank you both for accepting our invitation. It's really a pleasure to hear from you. So Paula, I guess Anna presents first. Is that by the program?
Yes.
Okay. So Anna, the floor is yours.
Thank you, Professor. I'll share my screen and then we will start, okay? Can you see it?
Yeah.
Yep. Good. So good morning for my colleagues here in Brazil and Latin America, and good afternoon for those in Europe. I'd like to thank the invitation to be here. It's really a pleasure. Thank you, Professor Barral, for the invitation. It's a pleasure to join you to discuss my favorite topic, which is precision oncology and precision medicine. I'm Anamaria Camargo. I'm a biologist. My research focuses on the discovery of biomarkers, predictive biomarkers, and identification of resistance mechanisms to anti-cancer therapy. The coordinator of the Molecular Oncology Center at Sirio Libanes Hospital. This is a private hospital in Brazil, here in São Paulo, but with a leading role in developing and especially in implementing precision medicine and more specifically precision oncology in Brazil. So it's going to be a pleasure to learn and discuss with you all. Thank you. Before I start, I'd like to make a few disclosures. The first one is that precision medicine and precision oncology is my favorite topic. I'm generally very, very optimistic about the benefits of precision medicine to human health, both at the individual level and also at the population level. And unfortunately, most of the information and conclusions that I will present reflect my own experience working in the field over the 20 past years. And this is because, unfortunately, there are not many systematic studies on this subject addressing the role, the implementation, the potential of precision medicine in Brazil. So I'll try to give some information I have from my colleagues working in the field and my personal view as well. So precision medicine, as all of you know, is a medical approach that uses, in addition to conventional clinical information, also genetic and molecular data to stratify patients and define the best strategy for disease prevention, diagnosis, and treatment. So this term became popular in the beginning of the 20s after the publication of the draft of the human genome sequence, and was actually officialized in 2011 in a report from the National Research Center in the United States about precision medicine. But in short, precision medicine aims to provide the right treatment for the right patient at the right time. So the benefits at the individual level are quite clear. I think they are mainly addressed through clinical trials, and these clinical trials have been very effective in showing that precision medicine is very important to define risk of developing a certain disease, to prevent this disease, to make the early and accurate diagnosis, improve treatment selection and efficacy, and reduce side effects. So this is easy to address using clinical trials. I guess the collective effects and benefits of precision medicine are more difficult to address, as they depend on real world data. And I think we can, at least in this forum, agree that it's reasonable, at least, to say that precision medicine has a great potential to improve population health, to reduce mortality and morbidity of different diseases, and most importantly, to reduce costs and delays in health assistance, especially in a huge country like ours here in Brazil. So I believe that the precision medicine in Brazil has a very promising future. So as I just mentioned, we are the largest country in Latin America. We have a population of over 240 million inhabitants. And actually, Brazil spends over about 8 to 10% of its GDP in healthcare assistance. So there is a lot of money spent in health assistance. And in 2019, the system actually spent about $55 billion in providing healthcare to the population. So we have the largest public health system in the world. We have universal access. All Brazilians are granted access to the public health system by law since 1988. But unfortunately, we can see in the reality much more of regional disparities, so developed and underdeveloped regions of the country. You have disparities in access, and most importantly, since we have a huge population, we have important delays in assistance. In addition to the public system, Brazil also has a proportion of the Brazilian population also have access to private health system. About 20% of the population, they can afford to pay additional health care. And what is evident in Brazil is that there is significant differences in access to drugs and medical procedures when you compare both systems, the private and the public health system. But the private system is also regulated by law and also by the Ministry of Health as well. So we have a long way to go in Brazil, and this is basically the big picture I can bring you from precision medicine in Brazil. So it's a reality for a small fraction of the Brazilian population mailing through the private healthcare system. Access to drugs and procedures, and especially reimbursement, remains a challenge here in our country. And this is leading to an increase in the number of lawsuits against the government and also against the private company of healthcare. So that there is a litigation trend that is increasing tremendously in the country. And this is something that we need to reflect a lot. But precision medicine is gradually gaining ground in the public healthcare. But unfortunately, and this is why I think these initiatives like we are talking today are very important, is that these changes here in the public sector are occurring in a very disjointly manner. They are based in immediate clinical needs, so we need, there is no planning actually, and in a still under development health innovation system. So innovation, this is not a very strong field in Brazil. We have much to learn. So most of the precision medicine initiatives we have in the country, they are still at the research level. This is not bad, this is very good, I'm a researcher, but we need also to think about implementation. And so far there is no implementation strategy in the country to grant access to precision medicine to the whole population. I'll show you some data after in the next slides. So the research initiatives in precision medicines are driven mainly by universities and a few public and also private hospitals like the one I work for, which are active in teaching, research and innovation. And what I think it's worth mentioning here is that these initiatives for precision medicine in Brazil, they benefit a lot from early genomics research projects carried out in the early 20s in Brazil. So I think I am a good example of this. I started working with genomics and cancer in a genome project that was financed by FAPESP in the early 20s. So currently we have a few initiatives in Brazil at the research level in precision medicine. They are mainly regional, involving very few institutions. For example, the EpiGen was the first precision medicine initiative in Brazil. It's from the University of Minas Gerais. The idea is to genotype individuals from longitudinal studies in Brazil. So this is the first attempt to characterize the genetic diversity of the Brazilian population. We also have a very interesting initiative that started in 2015 at the University of Campinas. This is led by Professor Isha Mendes. And the idea here is not to generate data, but to store and share genetic data related to human diseases. And this is interesting. They have a very nice platform so that you can submit and share your genomic data. This is an important initiative. I think we will learn a lot in the next talk about the importance and also the obstacles of data sharing. And this is an initiative that is done in collaboration with the Global Alliance for Genomic Health. So they follow the principles and they use the platform of the Global Alliance for Genomic Health. There is an important initiative at the University of São Paulo led by Professor Maiana Zatz. This is actually the first initiative to start whole genome and whole exome sequence of individuals from the Brazilian population. The project is focused on elderly individuals and patients with rare disease, but also it's a regional initiative at the research level led by researchers from the state of São Paulo. And only until last year, actually, we didn't have a very coordinated precision medicine initiative in Brazil. But last year, the government, the Ministry of Health, launched a project that's called Genomas do Brasil. The aim of this project is to establish bases for precision medicine in Brazil and foster its implementation in the public health system through sequencing the whole genomes of over 100,000 Brazilians. So this is a very ambitious project. It's still in its early beginnings, and unfortunately, due to the pandemics and also some budget cuts, we still don't have much progress in this project so far. But they exist. They start at least to discuss the subject, to provide sequencing data, and to start the discussion of data sharing and also ethical issues as well. So the obstacles of precision medicine in Brazil are many. Some of them are the same as in high-income countries. So regulation, the needs for data integration and sharing that we'll discuss in the next talk. The lack of cost-effective analysis to really address the viability of implementing precision medicine tools in the clinics. And we also have ethics considerations. So this is everywhere. This is observed. This is obstacles that we as human beings will need to face. But there are other specific obstacles for the local scenario, and these include limited research funding. We are experience a very hard time for research funding here in Brazil. We have a gap, an important gap in the ability to turn research into innovation. So the universities, they are not prepared, they do not have the tools, and most importantly, the scientists don't have the knowledge to move forward from research to innovation. In line with this, the national pharmaceutical companies, they have limited research and development activities. So they focus mostly in production and commercialization of their products. We lack infrastructure nationwide for genetic testing and also the use of new therapies like cell therapies. And this is especially important for precision oncology. We still have insufficient and untrained human resources, so we need to work with this heavily in the next years. And unfortunately, we have important delays in the registration of the drugs in devices. So especially if you compare Brazil to the US and Europe, some drugs that are already approved in these countries, they take in average 2 to 5 years to be approved here for use in Brazil. So what about precision oncology in Brazil? So precision oncology, I think, is the prototype for precision medicine. So the use of genetic information from the patient and molecular information from the tumor is key to define the management of cancer patient. So the use of genetic and molecular tests, as I will show you in a few minutes, are standard of care to diagnose and treat some cancer types like melanoma, breast, colon, and lung cancer. So this is already a reality in oncology, and we see that patients benefit a lot from this type of approach. So, and why this is like this, why precision oncology is so important in terms of precision, why is it the prototype for precision medicine? And this is basically because cancer is a genetic disease. So the basis of cancer are molecular alterations, and these alterations can be germline alterations that we get from our parents and define our cancer predisposition, but there are also thousands of somatic alterations that accumulate only in the normal tissue of the cells that actually promote transformation from a normal cell to a malignant cell. So molecular alterations are the basis of cancer, so they should be considered in the management of the cancer patient. So it's fundamental. So over the past 40 years, we learned a lot about cancer genetics and also the molecular basis of transformation. We have over hundreds of thousands of genomes, of tumor genomes that are already being sequenced. We are able to define the complete catalogs of alterations in humans in just a few weeks. Based on this knowledge, we have developed very important target therapies that are changing the history of the treatment of cancer, of a specific type of cancers. So just to give an idea, I think I have a number here. After 2011, there were over 100 new drugs approved for cancer in the world. So there is a lot of knowledge being translated into clinical management here. And this is also followed by the identification of an impressive number of diagnostic, prognostic, and predictive biomarkers, so that the development of genetic and molecular tests have also been astonishing. So we have over 75,000 genetic tests available for over 20,000 diseases, and most, I would say that over 20% of them are for different types of cancer. So this is an area where knowledge and application, they converge a lot. So basically in precision oncology, the pillars are the genetic and molecular tests, And these can be grouped in two main groups. Germline testing, they are made in blood and saliva. These cover genes, a few dozens of genes that are affected by mutations and are related with an increased risk of developing cancer during our lifetime. So based on these tests, you can use preventive measures for early diagnosis as well. You can do genetic counseling for the whole family, for planning, and also more recently, this information is being used to guide therapy as well. So germline mutations, for example, in DNA repair genes, they are now used as well to define treatment and the use of immunotherapy as well. But I think that the most important contribution of the genomics, cancer genomics, is the development of somatic testing. This is mainly due in the tumor tissue, but also can be done in the plasma. It's an interesting procedure called liquid biopsy. And these tests are used for screening and diagnosis, for molecular classification of the tumor, for prognosis, and especially to guide treatment. So this is actually the most important use of genetics and molecular tests in precision oncology. They are done to define therapy in many types of tumors, especially in the metastatic scenario. So this is the workflow. So you have the tumor usually diagnosed using imaging techniques. You extract tumor tissue, you extract nucleic acids, you do molecular analysis. Based on the molecular profiling, you can stratify patients, and then you can plan the treatment in a personalized or a more precise way. So initially there were a small number of genes that needed to be addressed to define treatment. I think the first example is an alteration that is frequent in breast cancer and involves the amplification of the oncogene ERB2. So these were one of the companion markers. Companion markers are tests that are used to define the use of a specific drug. So basically, patients with ERB2 amplification can use antibodies against ERB2 as part of their treatment. And then we start to improve treatment. There were new drugs, for example, anti-EGFR inhibitors are used for non-small cell lung cancer. Also, mutations in the codon 12 and 13 of the oncogene CARHAS. They are a very well-known resistance mechanism to therapy in colon cancer. But at that point, we just have a few genes to analyze. So genetic tests were not complex and they were not really high cost. So they were somehow affordable. But this is moving faster right now. We have currently over 500 genes that are assessed in molecular tests, and somehow they contribute to define therapy in oncology. So we now use in oncology very often cancer gene panels, addressing at least 100 of genes in a single assay. So cancer gene panels can be divided in small panels and also comprehensive panels. And of course, there are important differences in their use. For example, hotspot panels are used to guide standard of care therapy. So you have just a few genes, genes that are known to be related to success to treatment. So these tests are usually low complexity. You do not require huge bioinformatics analysis. They have a short turnaround time, but they are limited in the number of genes to be addressed, but also in the type of mutations and genetic alterations that are assessed. But you also have comprehensive cancer gene panels. They are used basically in the metastatic disease scenario. They have a high complexity. We are talking here about 500 genes being addressed simultaneously. These tests require a lot of bioinformatics analysis. They have a long turnaround time, over 2 weeks, but they detect all types of alterations, including alterations like tumor mutation burden and microsatellite instabilities that are now important to define the use of immunotherapy. So here in Brazil, as I will show you, the cost of these tests are not covered. They are not offered first in the public health system, and they are not covered also by the private health care system. So patients have basically to pay from their own pocket to make these analysis. And also, I think an important discussion at this point is the clinical utility of using these panels, especially considering their high costs. And the clinical utility is mainly defined by the prevalence of mutations in the target gene. So it depends from 20 to 40%. It varies from 20 to 40% of the patients, depending on the cancer type. Also, response rate is really important. You really need to use an efficient drug. And the access to the target therapy is critical. So it's not much help to know that you have a mutation if you don't have the access to the drug, or if you cannot afford the drug, or even if you don't have the health condition to use the drug. So the clinical utility is still under debate. But I think we will move fast here with improvement in knowledge in the developing and reducing the cost of the genetic tests and doing cost benefit analysis is a major issue and an urgent need here. So the obstacles in precision oncology in Brazil is basically the same that I have already described. Some are observed worldwide, some are specific to the country, and here I would like to emphasize the lack of infrastructure. So there are just a few centers in Brazil that can do next generation sequencing analysis and have the human resources, for example, for complex bioinformatics analysis. So we have to invest a lot in the infrastructure and also in training people. And we still have a long way to go. So this is to finish a brief picture of how is precision medicine, precision oncology in Brazil right now. So for example, germline testing for hereditary cancer syndromes are only available through the private healthcare. So you cannot do germline testing in the public system. And they only cover the most common syndromes like BRCA1, BRCA2 for breast and ovarian cancer, syndromes related to increased risk of colorectal cancer development as well. Somatic testing is available for a few genes like EGFR, ERBB2, KRAS through the public health system. This is a major achievement, but they are provided only in a few specialized centers. So we still need to work in this. But it's interesting because you can have broader access through the private health system as well. Pharmaceutical companies also offer these tests. sometimes, especially because they have interest in sell the drug, of course. Somatic testing, as I mentioned, using large panel is not available in the public health and not also in the private system, but they are offered by private clinical laboratories and hospitals in Brazil, like our hospital here in São Paulo. But also, I think I just want to finish that clinical trials are increasing in Brazil. This is a field that is rapidly expanding, and you can also have access to precision oncology through enrollment in clinical trials here in Brazil. So I think that was the picture that I would like to bring and discuss with you today. So thank you very much. I leave my contact for those that are interested in following this discussion. And I thank you very much for your attention.
Thank you, Anna. It was really a concise but comprehensive panorama of the precision oncology in Brazil. And we are going to leave the comments and questions for the end of the two presentations. So thank you very much. And if you could stop the screen sharing. And then we are going to move to Dr. Kirschner's presentation. And thank you again for accepting the invitation, and the floor is yours.
Sure, thank you very much. Yeah, during the next half hour or so, I'll will provide you a sort of high-level overview of the of an EU project which I'm coordinating, and yeah, as Professor Barrell already mentioned, my current occupation is being a scientific officer, and I mainly work for the German Ministry of Education and Research in this function, and we are basically developing strategies with the BMBF and also fund projects where the BMBF is the grant provider. And but we're also active on a European level, and here my unit which I'm working for is very often associated with and applies for coordinating and support actions, so strategic projects. And the project I'm presenting you today is one of such strategic projects, and I divided basically my talk into three parts. I will give you a brief project summary, and then I present you some project outputs, and then I will concentrate on a sort of milestone of the project, which is an Iso Technical Specification. And I hope that you might be interested in also working with us, basically improving this Iso Technical Specification. So EU stands for PM, is basically a coordinating and support action. And we're not a research project, we are purely a networking and strategic project funded by the European Commission. And the project runs for 3.5 years. It started in 2019, and we recently got an extension of the project. So the project will run until end of June in 2022. We have a budget of roughly 2 million euros, and the consortium consists of 6 partners from 8 European countries, and coordinated is the project by Forschungszentrum Jülich and by the project management agency for which I'm working for. So this slide gives you basically an idea, and really overarching idea, what our central question is in the project. And we basically want to know, where do we get from data to knowledge with the help of in-silico modeling approaches, so meaning computer-based models. And we're clearly addressing all these different types of data summarized here in this left part of the slide basically all clinical derived data, clinical registry data, molecular data, pre-examination, and so on and so forth, but also lifestyle data. And we are interested in suitable ways and best ways to harmonize and integrate these data for predictive modeling approaches that have an applicability in the clinic in the context of personalized medicine. And we are focusing our work on recommendations, guidelines, and standards for data integration and model validation in the context of these predictive model simulations. And what we do is we provide technical, feasible, and an ethical legal frame, but also flexible guidelines for data sharing and data governance. And our target communities are European collaborative research projects, but also funding organizations, national funding organizations, but also funding organizations, for instance, on a European level, such as the European Commission. So this gives you an idea, this slide gives you an idea of the basic structure of the project. We have basically 4 different work packages, whereas work package 1 and 2 are complementary to each other. Work package 1 deals with all different kinds of data sources, and work package 2 deals with the corresponding in silico modeling approaches. Then we have a legal and ethical framework established by work package 4 and work package 3, and work package 4 works on data access and governance, and also awareness. And the specific project outputs are basically assessments, so state-of-the-art reviews, recommendations, guidelines, and standards. I'm coming to this a little bit later, and dissemination activities, such as the seminar we're in today. So now I will present you some selected project outputs for the corresponding work package 1 to 4, starting with work package 1, which is the work package that deals with different data sources and models in personalized medicine. And we're currently working on a report on a EU-wide mapping report of data sources that are relevant for personalized medicine. And this report is basically based on a comprehensive online survey with roughly 90 questions. And we addressed more than 70 respondents and asked specific questions about their data sources and standards, modeling methods and standards, and how these respondents got access to their data and consent. And yeah, this is a work in progress, and the report is at final state and will be submitted to the European Commission soon, and also published as a manuscript, hopefully also towards the end of the year, beginning of next year. So I'm now coming to sort of the central work package of EU stands for PM. This is work package 2 that deals with our main focus, which is the in silico model approaches. And here at the in the first half of last year, we assembled a white paper. We wrote a white paper that discusses the most relevant computer-based modeling approaches for personalized medicine, which are the classical cellular systems biology models, models for risk prediction for common diseases, but also models for disease cause and therapy response predictions, pharmacokinetic, pharmacodynamic models, and artificial intelligence models. And we developed a first set of recommendations for data integration and model validations for these model approaches that is backed up by a collection of use cases. And this work has been done by Catherine Collin from the University of Copenhagen. And in 2020, we also published a paper from a workshop we did with the Combine community. And this paper contains a first set of more or less sort of common recommendations addressing funding organizations such as the European Commission, but also collaborative research groups, journals, as well as policymakers. And we are currently submitted a review article that exactly deals with these different modeling approaches I presented earlier. And here we provided a more detailed description of these clinical relevant modeling approaches, and also for computational concepts for patient stratification in personalized medicine with the help of these modeling approaches, and but also how one could foster discovery, diagnosis, and therapy in personalized medicine with these models. And we also screened the literature and provided a sort of actual overview summarizing a collection of use cases where these modeling approaches are being applied currently. And we discussed challenges and also then, of course, provided recommendations how to use these modeling approaches in the context of data integration, harmonization, and model validation. And so this is also a milestone of the project. We are collaborating with Iso, and we currently submitted an Iso Technical Specification. And this has been accepted as a new work item last month. And for those of you who are not familiar with the ISO deliverables, you all might know an international standard, which is the highest ISO deliverable. Is one of the most normative documents with a high binding force, but sort of right below the international standard is an ISO technical specification for a field that is currently under development. And this ISO technical specification is also a normative document, meaning it has a certain binding force, and it can eventually be transformed and republished as an international standard. And yeah, so I'm coming to this a little bit later in my talk, where put some where give you some more information on this ISO technical specification, and also some ways how we probably could interact and collaborate together. So we currently working on another article, which which is best described as a sort of practical implementation guide for clinical driven projects to realize personalized medicine. And this is an article that specifically deals or specifically discusses challenges, hurdles, and problems that clinical driven projects in personalized medicine have. And we provide sort of solution and recommendations how to overcome these challenges and hurdles in a clinical driven project in personalized medicine. So I'm now coming to the legal and ethical frame of EU stands for PM, which is covered by Work Package 3. And currently we finalized 2 reports dealing with a legal and ethical review of this in-silico modeling approaches I showed you earlier. And these 2 reports discuss in detail the legal and ethical framework with regard to consent, the general data protection regulation, and patient rights. And the other report discusses ethical, legal, and sustainable avenues for harmonization and integration of big data for in silico models. And if you're interested, these reports can be found on our website, and you can easily download the PDF files from the websites if you're interested and you wish to read them. I can highly recommend them because it's a completely different view from legal and ethical standpoint on this complex topic of data integration, patient rights, and the proper legal and ethical framework of this. So, and this work is done by Mette Hartlef from the University of Copenhagen again. And currently, work package 3 has published another article on research ethics and personal data protection. And if you wish, you can also find a download link on our website and have a more detailed look on this specific topic. And this work has been done by Eugenius Jeffernas from the Vilnius University. Another very interesting or critical topic we're working on is data governance and data access. And this is being done in work package 4. And here we developed a new harmonized data access agreement. And this is basically a template, a harmonized template to obtain access to controlled access data, meaning those data that are deposited, for instance, in the European Genome Phenome Archive. And this template provides a better data flexibility across collaborative research projects. It is fully GDPR compliable, and currently it's one of the 2 default templates that can be that are available for download on the European Genome Phenome Archive. And here, if you're interested in the details and a little bit more in the background of how this was being developed and how and the current challenges associated with access with data access for controlled access data, you can find some more information on our website again. And this work has been done by Stamattina Liossi and Stefan Beck from the University College London. So I mentioned this already earlier. We work together with Iso, and we established during the course of the project, we established 2 liaisons with Iso technical committees. This is the Technical Committee 276 Biotechnology and the Technical Committee 215 Health Informatics. And currently under review is our ISO technical specifications here in the Technical Committee 276 and the Working Group 5 Data Processing and Integration. I'm coming to this a little bit later. So now I would like to focus a little bit on the ISO technical specification I mentioned earlier, because I think this might be also of interest for this group here. And so why did we do this? Why did we work together with Iso and try to develop a specific Iso deliverable? And as I mentioned earlier, we are basically focusing on data integration and model validation in our project in the context of computer-based models. And the target communities of funders and collaborative research projects. And currently, the situation is that there are no broadly accepted standards for health and disease data integration and harmonization for predictive modeling approaches in the context of personalized medicine. There are many grassroots, but no, not really binding and formal standards. So the area is basically highly fragmented. And as a result of this, there is a clear need for common standards and for harmonized strategy for data integration to enable computer-based modeling in personalized medicine, but also for sustained use of data in clinical and health research based on the FAIR guiding principle. And there's also a need for broad adaptation of these harmonized health data and standards in research that are backed up by a sound legal and ethical framework I also summarized earlier to you. So, and so on this slide, you see how the different publications and papers and articles are coming together. And so we basically have our white paper I mentioned at the beginning, then we have the review article and the Iso Technical Specification. And hopefully this work will pave a little bit the way towards broadly applicable standards for predictive modeling approaches in personalized medicine. And we really hope that funders as well as collaborative research projects might benefit from our work here. So now I'm providing you some details on the Iso Technical Specification so that I can have an idea how such a document or how this document looks like. Starting with the title, and the title is the Recommendations and Requirements for Predictive Computation Models in Personalized Medicine Research, and we have part 1, Guidelines for Constructing and Verifying and Validating models. So this basically covers everything I mentioned during the last 20 minutes, basically. And the scope is, I'm not reading all through this. I just want to highlight some of the most prominent words and phrases here. So it's computation models constructed for research, requirements and recommendations, challenges regarding data input, and as well as verifying and validating of models. So these are the most important keywords when you think about this Iso Technical Specification. And we divide, so this is the table of contents, and we divided the Iso Technical Specification basically in 4 different parts. The first part dealing with the different modeling approaches, and the second part dealing with data integration into computer models, and then model formatting, model validation. And the fourth part deals with the legal and ethical requirements for these different modeling approaches. So I'm not going any further, not to probably to bore you with too many details. I just want to give you sort of a brief overview of the content of the Iso Technical Specification. And now I'm coming to the end of my talk, and as I said earlier, E stands for PM is a networking and strategic project, and we basically an open network. We are happy to provide information and also collaborate with other relevant projects. Here's a brief summary of the contacts we established so far, and during my last slide, I would like to thank the key players and also provide you again a little bit overview of the key players and their specific tasks, divided here into different groups. We have on board in EU stands for PM 5 ongoing large Horizon 2020 projects that deal with different aspects of personalized medicine and computer-based models. And we have for our standardization work, we have as a quite prominent partner is the DIN, the German Institute for Standards, and as as representative of Iso in our consortium, we have quite an extensive experience for data and models with Alex here on board, and different unit groups from university that specialize in data and models. The legal and ethical framework, you saw that already, is provided by University of Copenhagen, of University of Oxford, and Vilnius University, and we have support by a regulatory agency, the Federal Agency for Medicines and Health Products in Belgium. Yeah, and so this is really my last slide, and with this I would like to thank the European Commission for funding. EU stands for PM, and you for listening to my talk, and I'm happy to take questions. Thank you very much.
Thank you, Dr. Kirschner, and it was really an impressive work. I mean, performed in several critical aspects for really promoting this data integration and with the fair use and ethical and legal aspects considered beside the only technical. So it's really interesting. I mean, it's possible that we will have some further talks and meetings on how we can deal with some of these documents in Brazil, and what we can really, I mean, use of this so well conducted study to really, I mean, promote the data integration is critical for us in terms of national database, but it's even better if you do that already thinking in terms of international kind of standards and how to make easier to integrate data from different regions. This will be critical not only in precision medicine but in several aspects of I mean, health research. So thank you both. It was really, I mean, very good panel into different aspects of the, and if somebody has some point to raise or question.
So I have a first question. It's very much linked to the comments you just raised, Manuel, for Dr. Kirschner. Regarding the Iso Technical Specification, if I understand correctly, the idea is that it could be globally applicable. So anybody anywhere working on these topics could apply eventually this this technical specification. Is this correct?
Yes. Yeah. So the current situation is that we submitted the Iso Technical Specification as a so-called new work item in July to the Iso Technical Committee and the Working Group 5 I mentioned, and then in last month it has been reviewed, and then sort of it's basically a pre-reviewing process, and last month it has been accepted as a new work item. So meaning that it's currently under official review in this Working Group 5. And this reviewing process will take up to 2 years. And during these 2 years, more or less consensus that the document is reworked, reviewed, edited, and improved. And once all the members of this Iso Technical Committee basically agreed on the content, then it's being published as a technical specification. But during this reviewing process, you always have the chance to provide input, and this is also what I would like to discuss with you. We could basically go into an internal reviewing process with you. I could provide you the document, and if you're interested, you could send it back to me with your comments, and then I would basically during the next ISO meeting discuss these comments with the ISO working group, and if they agree, then they will be implemented. So this is more or less, this is also an iterative process where the document is improved over time.
Thank you. That was my
Dr. Oh, sorry, Paula. Go ahead.
No, I was just going to say that was my follow-up question to better understand how the technical specification keeps getting updated, because I know it's a very rapidly evolving field. Sorry, Manuel.
No, it's okay. I was just going to mention that Dr. Oman is raised the hand. So.
Yes, thank you very much. I have one question. I think this is the internal review is a very good idea, because unfortunately the ISO documents are not openly available. So it's difficult if you have no license to get this information, and to distribute it to the group is really excellent, I think.
But
Thank you.
Can what you can do is it's the same what we did in EU stands for PM. We applied for an liaison with ISO with a technical committee and a working group. This is also what you can do, and if you are interested in working with ISO, please let me know, and I'll establish the contact so that you can also be you could for instance be an expert in this specific working group and sort of officially under ISO work on these kind of documents.
Thank you.
Just let me know, drop me a line, and I'll establish the contact.
Well, I was going to ask Anna if she's aware of any movement from Paulo and FAPESP on the efforts of data integration. Everybody heard about this during the COVID, of sharing data between institutions, and Sirio is part of that. But I mean, the initiative, FAPESP initiative on data integration, I'm not really aware how it goes, and of course it's not your area directly. But if you have some news, if you could share, it'd be nice.
Yes, I think this is a very important initiative that FAPESP is undertaking. It's led by Professor Isia Mendes at Unicamp. And the idea, I think, goes very well with what has just been described to data sharing. So basically, they are building a repository so people can share sequencing data and also clinical data as well. So, but again, this is a very regional, university-based initiative. I think we need to move next step for something more structured, like we just have here, that would be coordinator. The problem, I think, is not much of the genomic data, because especially scientists, they know how to handle and share data, right? I think the problem here in Brazil is medical records, because we'll still have no electronic medical records, especially in the public health system, but also in the private hospitals as well. So for us here in São Paulo, in Sirio Libanes, which is a very good hospital for Brazilian standards, and we'll still face problems with the clinical data. So electronic medical records should be improved in Brazil if we really want to share data in the world of precision oncology.
Yeah. Any further questions or comments?
It's me again. Just I had a question for Dr. Anamaria, because you mentioned, so you feel there's an important gap in Brazil between research and innovation. And I don't know, in your opinion, where should efforts focus first, let's say? Is it more of a training issue where it's important to start by building more capacity among the young scientists, or is it more of a resources and funding issue, or is it more of an infrastructure issue? If you had to decide where the efforts should go first. You're on mute.
Sorry, I think that you listed the three main points, right? Training, funding, and I think I would put emphasis in funding, because we have people. I think the universities, they are starting to have like startups and so on, but we need funding. I think funding would be the first choice right now, because we have infrastructure, we have good people and people that are willing to learn. But right now, I think we have very few startups in the country, and the mechanisms of funding are not good. I mean, I would go for funding.
And it's not only good, not only because it's very small, but because it's also extremely bureaucratic to use and difficult to use, even when it's funded. So we have both problems with amount of funding, but also with dealing with funds and administrative aspects.
That's right. Yeah, that's critical as well. Good point.
So, Maria?
Yes, sorry, I have problems with my camera, but I would like to ask to Marc if there is, well, my question will be, have in-silico models some specific ethical or legal issues different from the usual ones of personalized medicine?
Not that I'm aware of. I mean, the big issue is always when it comes to the patient-derived and personal data and the content question. But if you would really have some more detailed and in-depth look and discussion, I can really highly recommend the two reports that have been published by the work package 3 that dealing with the legal and ethical issues of these approaches.
Okay, thank you.
So I would, I guess we can begin to close. And thank you again, Anamaria and Marc Kirschner, for very interesting and attractive presentations with a big picture in each case of the situation in Brazil and the data integration in Europe with possibilities of be of very good use abroad. I mean, in different countries also. And I would like also to thank Flavia and Paula for putting this webinar together, because I mean, I'm really coordinating here, but they did all the work behind the scenes, and so it's really, I mean, thank both of them for the in the project and all the support of EULAC and Maria and the people there in Spain that also is elemental for putting everything to work. So thank you a lot, and we are going to have probably only next year some other activities, and I will let you know. Okay, thank you. Bye bye.
Thank you.
Everyone.
Bye bye.
Pleasure.
Thank you very much. Bye.
Bye.